Dear panelists, Ben Herbert and Jacob Hanna, dear organizers and participants of the Marsilius Summer Academy, dear colleagues, ladies and gentlemen, a very warm welcome to all of you here at the Alte Aula. On behalf of the Marsilius Kolleg, I'm delighted to open this very special event here in these holy halls of the Heidelberg University. My name is Friederike Nüsse. I'm a professor of systematic theology and one of the two directors of the Marsilius Kolleg, which I have been leading together with my colleague Michael Boutros since 2020. The Marsilius Kolleg is the interdisciplinary institute for advanced study of this university. Since 2008, it has dedicated itself to bridging the gaps between the different fields of academia, especially between the sciences and the humanities. Every year, we appoint a group of up to 15 scholars from all areas of the university to meet weekly to discuss and develop their interdisciplinary research projects. The range of topics in each class and over the years has been extremely broad, ranging from climate change, digitalization, artificial intelligence, to questions of anthropology, social cohesion, or theoretical issues such as complexity and simplicity. Over the years, the Marsilius studies as a platform for our students and doctoral candidates, the Young Marsilius Fellowships for postdocs, and the Nature Marsilius Visiting Professorship in Science Communication have been added as an important pillar of the Marsilius Kolleg. Since 2009, the Kolleg has regularly hosted so-called Marsilius Academies, one-week workshops for excellent doctoral students from all over the world, working with outstanding speakers on an interdisciplinary and socially relevant topic. After a break of almost 4 years, we are particularly pleased and proud to present the 15th Marsilius Academy this year under the title At the Limits of Life. 20 doctoral students and 9 excellent speakers from the life sciences, engineering, social sciences, and humanities have been discussing the changing limits of life since Monday morning. We are very happy that we are able to overcome boundaries of the scientific landscape in Heidelberg and win the European Molecular Biology Laboratory, EMBL, and the Max Planck Institute for Medical Research as cooperation partners for this summer academy. With today's event, we want to involve the broader public, all of you, in the discussions of the Marsilius Summer Academy 2023. For a long time, the boundaries of life seemed predetermined and therefore immovable. In recent years, however, research in the life, natural and engineering sciences has challenged these boundaries at some critical points. Tonight's panel discussion takes up one of them, which has received a lot of attention in recent months, the question of whether or not the beginning of life remains inaccessible to humankind. The public debate on the ethical, legal, and social implications of synthetic human embryos has only just begun, and we are all here to be part of it tonight. I would like to thank those who made this event possible. Our distinguished panelists, Jacob Hanna and Ben Herbert, our 2 fellows and moderators of tonight's event, Thorsten Moos and Jochen Wittboth, the team of the Marsilius office, in particular Niels Schütz, our cooperation partners from and the Max Planck Institute for Medical Research, the Heidelberg Mannheim Health and Life Science Alliance, and the Cluster of Excellence 3D Matter Made to Order, who generously supported us with some additional funding. Last but not least, thank you all for joining us tonight. So let me hand over to the moderators of this evening, my esteemed colleagues Thorsten Moos and Jochen Wittbrod. Thank you very much. The floor is yours.
Thank you very much. Thank you very much. A warm welcome to all of you. My name is Thorsten Moos. I'm a professor of ethics at the Theological Faculty of Heidelberg University. And together with my colleague Jochen Wittbrod, professor at the Center for Organismal Studies, Heidelberg, I have the honor to chair this event. There is something going on in the field of embryology. A few months ago, 2 research groups declared that they were able to grow living structures in the lab that closely resemble human embryos, including their surrounding tissues. Jacob Hanna's Nature paper on this was precisely published last week. Thanks, Jacob, for this timing. These embryo-like structures were grown from human cells, yes, but not by using egg and sperm cells, as in the case of in vitro fertilization, but only by using stem cells cultivated under different circumstances. Synthetic human embryos, they were named by some. So what does it mean that there are now entities that look like embryos, that have very similar biological characteristics as embryos, but that have a completely different history and probably future than embryos have? What does it mean for our understanding of human reproduction? What ethical, legal, political issues are connected to that? What future perspectives arise? With these questions, we really are at the limits of life, and we are so glad to be able to discuss it with all of you.
Thanks, Thorsten. Also, very, very warm welcome from my side. As Thorsten indicated, Jochen Wittbrod, developmental biologist, so not a theologist. So maybe I take the occasion of being in that old hall to refer to something our rector always likes to refer to, namely that this is the hall where debates were held. There was thesis, antithesis, and eventually there was a synthesis. So it's not a negative connotation, but rather something very, very positive. So imagine, we know a lot about the moon. We're not that much traveling the ocean and the floor of the ocean. We know a lot about, in molecular details, how an embryo develops when it comes to Drosophila and mouse. Well, we don't know that about humans. Wouldn't it be cool, and not just cool, but really very rewarding in multiple respects, on the one hand for basic research, on the other hand for translational applications to understand how a human embryo really works without need to interfere with human embryos. And I think that is the stage at which Jacob Hanna and his team are playing. And we had him the full day, and I have to say I was so excited. I thought, well, there's many questions, but he didn't answer them all. But the way he was openly phrasing them made me extremely confident that we're going to be excited. You will be excited at the end of that day. And looking at the audience, I'm really super, super happy that this is not the usual Alter Aula audience. It's young, very young. So I'm very glad that the future is here and really wants to hear about potential new approaches and contribute and participate actively. So let me briefly introduce Jacob. He's going to present the first thing. Jacob is an MD, PhD, got his degree in Israel and was microbiology and immunology. After that, he moved on. That's the normal way of a scientist. Looking around, you most know what and how scientists move. So he went to the US, he went to Boston, and went to learn about stem cells in the key, well, at that time, the key lab of stem cell biology. And after a few years of postdoc, he returned to Israel, to the Weizmann Institute, where he started his own group and started developing from that point of status quo stem cell biology into new areas that we get extremely excited about. And I don't want to talk about what he's doing. He should do that himself. We're just glad and excited that all the timing worked the way it did. And we're looking forward to your presentation, the discussion afterwards. And as you all know, it's not going to be over after 90 minutes. We're just moving on to discuss in smaller groups in the bel etage afterwards. And without further delay, Jacob.
Thank you. Thank you very much for this overly kind introduction, and really nice to be here in Heidelberg in this really beautiful hall. We'll start, yes, with kind of a short presentation. We want to just make sure everybody really sees the science and sees what we're talking about when we talk about synthetic embryo or embryo models. Before we dig deeper as well, and then my colleague Ben Halbert as well will give his short introduction, and we proceed. So, as you know, stem cell field has been, this dream of trying really finding body parts and organs for transplantation has been a very long goal, really. If you can see this Cranach painting from the 16th century, the Fountain of Youth, where you can see the older come and bathe in the water of the fountain, go out on the other sides, much healthier and younger. And you have the more modern version of the Times about this. Really, it's not only for the patients and ills, but also we want to improve our quality of life. We want to be healthy, have good elbows and knees when we are 80 and play tennis and so forth. So this Fountain of Youth really, the image is looking how embryo development, and our development all starts with a single cell, coming to a 64-cell stage, which is called the blastocyst. Implantation happens, and from that, all tissues of the embryo develop. Unfortunately, 40 years ago, first in mice, 20 years ago in humans, scientists were able to take cells at this early stage of the blastocyst and grow them in the dish indefinitely. These are called embryonic stem cells. They keep their quality, and in mice, if you inject them back actually into the embryo, they can make an entire mouse out of it. So what is the goal really of the stem cell research overall? The main goal is really using the potential of stem cells that they can make every cell type and try to provide cells that are customized, matched for patients, really replace organs. But as you know, the field developed, we want to make stem cells from the patient himself, that they can match the DNA quality, so there will be no rejection, even no need to search for donor. Here came a breakthrough from Yamanaka in 2007. He got the Nobel Prize for it, that he showed he can take any adult cell or skin cell or blood cell and bypass using eggs or cloning and so forth and directly revert it back into an embryonic-like state. And these are called iPS cells. And just to show you how this looks, these are the fibroblasts, the skin cells. You can see these colonies emerging. And then after that, you can end up with these shiny colonies, and these are the iPS lines. And you can see this in live imaging over 2 weeks. Excuse me. You see the colony of iPS cells emerging, and we can do this from any age we have. So we can make iPS cells, we can make ES cells in mouse and in humans. And really, just to show you what is kind of a dream scenario in a way, something we showed in principle in mice, that we can take mice that have a genetic mutation like sickle cell anemia, which is a mutation that makes the blood cells break, basically. And in mice, we can take skin cells, tail cells, grow them in the dish, express this Yamanaka factor, make iPS cells that are genetically identical, replace the mutation because we have to correct them, and then we have to differentiate them. We cannot, in general, just transplant embryonic stem cells and expect that they will learn from their neighbor cells. We have to differentiate into the specific cells. In this case, we made bone marrow-like cells, transplanted it back in the mice, and we saw that 80% of the mice really recovered. So that is nice. But really, the big challenge for the field, which is a bit surprising, is that differentiation has proven very, very difficult. Both in mouse and humans, when we try to differentiate cells into specific cell types of, like, let's say, mature liver or pure liver cells, we still cannot do it for most cell types. But I don't think that is impossible. Really, if we want to make differentiation better, we need to imitate the embryo, because the embryo, that's what it does. It's the best organ-making machine. However, we have a problem in humans. We don't know what we need to imitate. Because, in general, in mammalians, and that's where we start, where the stem cells start meeting embryology. Now this field is being called stembryology, because we can grow the embryos until the 64-cell stage, and then implantation into the uterus has to happen, both in mouse, in humans, all mammals, almost. And then quickly, in the first 4 or 5 days, you can see that the embryo moves from a ball of stem cells and makes all its organs in 5 days. In humans, this happens in the first 5 weeks. So the uterine barrier, the problem is that this happens very quickly. The embryo is very, very small. It's inaccessible, too small to look at. And of course, when you move to humans, you have technical and ethical barriers. At this stage, of course, women usually don't know they're pregnant. And even if for some reason we get some sample here or there, this is good to make some atlases and sections, but to study biology, we need hundreds of thousands or millions of biological samples. And this is never, and justifiably never, gonna be impossible always with the human cells. Therefore, we started thinking as a field, are there ways to bypass what happens in the uterus and make a model from stem cells and see if we can recapitulate some of these stages. And we started this, first we said in mice, of course, because everything works first in mice, we know it much better. And before trying to make embryos from stem cells, let's first learn how to grow a regular embryo, a normal embryo outside the uterus. And very briefly, we started building or reviving basically devices that can grow mouse embryos for a prolonged period of time. You can see here on the right this incubator with a wheel and a glass bottle that makes humidification, a gas regulation, which is kind of regulates a lot of the gas, the pressure, the temperature, so forth. And it's all mixed and circulated back to this device. And you can see here we start with day 7.5 mouse embryos. And when the system is working, each one is growing in its own liquid, which has its own nutrients that are calibrated. And we can see here, we can go from day 7, and 1 day later, you have the yolk sac with a heartbeat. 2 days later, you can see the embryo is turning with the head and the feet. 3 days, bigger heart with blood. This is the fetus blood. It has a placenta, but it's not attached to the mother. And this far, we described up to day 11. Now, this was a very important base, and it just already shows you as what are the potential that we can do. For example, we can watch things live at certain points and see how this process called gastrulation, where we really make the beginnings of organs. You can see how this primitive streak and the somites form and the head form, and we can really start watching things in high resolution live. And again, we're not only trying to make cells for transplantation. I want to say our knowledge also and intellect is very important. Gastrulation, Lewis Wolpert has a famous quote which says, you know, the most important day in our life is not when we marry, it's not when we are born, but actually it's gastrulation. Because if we don't gastrulate correctly, we're going to have developmental malformation. And with this progress, really, we're able to show that in mouse embryos, we can grow embryo for about 6 days outside the uterus, which is about a third of pregnancy. And these are the critical parts. These are the parts where it starts creating all its organs. And really shows that the embryo, in a way, does not necessarily need the uterus, and that it make its own shape without the uterus. And now that we had conditions we know can support mouse natural embryo growth, we went to look at, can we take now stem cells and put them in this device and systems and see what happens. And we started using stem cells that are very early. We call them naive in mice. We take them into 3 parts. One, we push it a little bit to become a trophoblast, which is the placenta. 1 nudge to become the yolk sac. After 24 hours, we put everything together and mount it on the systems and the device. And this is really what I think is powerful. You can see the 25 cells start growing, just looks like a clump. But at day 4, you can see that you're getting an embryo-like structure similar to day 5. 1 day later, you can see the similarity. This is very sophisticated. Day 7, you get the yolk sac suddenly with the embryo-like entity inside of it, and we can reach day 8.5. And really, if you just look at it again, we do this using the same device, the same conditions, the same media, but except that these structures now are made from in vitro stem cells alone. If you look at these entities, you can see that they are developed, they start past gastrulation and have structures like the brain, the beating heart and valves, the allantois, which is going to become the umbilical cord. If you look at the yolk sac itself, you see these clouds because the first blood cells actually are formed in the yolk sac. So just briefly say that if we compare natural embryo with a synthetic embryo, we can see the patterns progressing correctly, and we end up with a different compartments such as the heart in pink, or the head, or the neural folds, what's called, both of them without us giving the morphogens, that this is self-organizing. But I want to highlight, and this is relevant to discussion, if you look at the synthetic embryo at the top, it's not fully normal. It's never fully normal. The neural tube is very convoluted. The heart is often too big. But on the other hand, you clearly have the map and the architecture of an embryo-like entity. And the last section is really trying to do this with human stem cells, and this is work of a very talented student and a postdoc, Vlad Bondarenko, who's trained here at Embryos. And really, here we had to do some adaptations. We have used these human naive cells, but you can already see from the picture, the scheme of the human embryo at day 14, it's very different from what I showed you from the mouse. And we had to think which cell types we need to make. I wouldn't get into the details. There are lineages that in humans are not found in the equivalent part in the mouse. But if we adapt this protocol and we can really get this to work, we start with 120 human naive cells, meaning that they're really the earliest state in development. And the first 3 days, not much happening, but day 3 and on, you can start identify there are cavities and different compartments, which we can really validate and annotate that this looks what is described in textbooks. Best thing just to show this by a movie. You can see here these are kind of three-dimensional movies, and you can see at this day 14 equivalent set, you have the trophoblast, the pink on the outside. And then when you look inside, you're going to see the yolk sac in the yellow underneath it. And then you have this layer of flat yellow with the blue. It's called bilaminar disc. It's a human, not a mouse. And on top, the rooftop is called the amnion. So again, just the point is that you're really getting three structural self-organization of the different compartments. And this is when you kind of stay in every section and go through it. And this is what it looks like. And let's show an example that we can really get the development in fine detail. So for example, if you look at the yolk sac, so on the right here, we have this atlas of human development from the 60s. We didn't use any human embryo material newly derived in this study, but we can really learn from these atlases. And you can see how, when you go through the video, the composite, you can see the yolk sac, and you can see the epithelium on top is columnar, versus at the bottom, it's squamous. So really, you're getting fine architecture, not only compartments, but you're starting to get these small details, and that really this is driving, that this wasn't even before in the cells that we introduced. And just a last example, I'll show you about the trophoblast. If you look from the outside, we're starting to get features that are advanced trophoblasts. So in humans, you have what's called lacuna. These are like, you can see that you're looking inside a window kind of thing. And this usually gets filled with blood inside the uterus. And you can see pregnancy hormone secretion, the green color, really showing that we're recapitulating the architecture of the embryo at these stages, although we can see that they're also not identical and the efficiency is very, very, very low. As I said, why, just briefly to touch, why are we doing this overall? We are trying, in the first 5 weeks of development, a lot of the pregnancy failures happen then, a lot of the malformations develop then, and a lot of the drug toxicity happen then. For example, today we cannot study drug effect on pregnant women because it cannot be tested there. But we can see, for example, in mice here, we show this, we can take these embryos and add a drug called valproic acid, which is known to cause brain defects. And on the right, you will see that the brain, we immediately see the brain defects that we know. So really giving us a little bit preview, perhaps, or one of the things we want to try to do in humans, look at development of human embryo models as they progress and test drugs and so forth. And another thing which we ask, we also want to use them for learning how organs form, which genes are important, and maybe some of the cells in there are precious for transplantation or testing. So what I'm drawing a scenario now, maybe one day for a certain indication, maybe we think of somebody has a leukemia and about to die because he cannot find a donor of bone marrow, maybe we can take a skin cell from him, make these naive iPS cells, make these embryo models up to about day 40, and take bone marrow cells, in this case blood cells from the liver, and transplant them, which are identical to him, no need for donor eggs, no need for aborted material. So this technology might have some advantage of making high quality cells. Of course, we're not blind to a lot of the questions, the ethical questions that are very justified. And we've recently kind of made this famous painting about the treachery of images by Matisse, and translating by saying, this is not a mouse embryo. And the idea is to raise the question, is it or is it not? And what is the barrier, which we actually don't know about really. And we're trying to engage the public to hear more and learn about ideas and concepts and concerns. And I'll finish just this last slide by saying that we are already facing challenges as we do the research. What should we call this? Should we call this a synthetic embryo? Should we call this an embryo model? Should we, when we call a structure, yolk sac or yolk sac-like, am I sure it's exactly like a yolk sac inside the human embryo? Of course not, because actually we don't have much controls, as I told you before. So there's a lot of dilemmas when we make statements about this. And we don't want to overstate, but on the other hand, we want to mislead. So it's not easy. And the other example, when we talk about complexity of structure, should I call this complete? Should I call this bona fide? How do the people interpret it? Do I know what's the potential of this to develop? Actually, I don't. And a lot of dilemmas where how does this get perceived by the audience? Should we actually engage with media very early before things are published? Or should we actually do this very carefully with explanations, with videos to make sure that this topic is really sensitive and we must really be careful that this doesn't lead to misleading information. So a lot of dilemmas, just even at us personally, the scientists dealing with this. And with that, just to finish the introduction, just to introduce the topic, and thank you, and look forward to discussions afterwards.
Thank you so much, Jacob, for this intriguing introduction. And now we want to discuss, and we need two to make up a discussion. And I want to introduce, probably not as an opponent, but surely as someone with a different perspective, a perspective from the social sciences, Benjamin Holbert. You know his family already. Now let's come to him. He's an associate professor at the School of Life Sciences at Arizona State University. He calls himself, in the preface of one of his books, a disciplinary misfit, and this is why he's a perfect fit for us tonight. He was trained in science and technology studies in Stanford and in Harvard, with a focus on the history of precisely our topic, the history of the modern biomedical and life sciences. And he's looking at this intersection of bioethics, science and technology, and political theory. How do democracies relate to science? How does something like public reason relate to science? There's only one book I want to mention and highly recommend among his works. It is called Experiments in Democracy, Human Embryo Research and the Politics of Bioethics. Ben, we are glad to have you here. The floor is yours.
First of all, let me thank our hosts and organizers. It's really a pleasure to be here and to have participated in the last few days of intensely rich discussion. It's also an extraordinary pleasure to be in this beautiful room. I don't think I've ever spoken at a lectern that looks so much like a pulpit, so I will do my best to respect the difference, especially in the land of Max Weber. So I have the challenging task of making moral sense at the limits of life. I will spectacularly fail in this task, but I hope to put a few issues on the table that can be the basis for collective discussion, which, to anticipate my conclusion, is in effect what I think is the most important task before us. So Jacob has introduced you already to the extraordinary work that he's done. I put up this headline. This is an article from The Guardian from just a few days ago, just to point out how sort of tortured the task of writing a headline for this domain is, not least because this question seems to be emerging as the fundamental question, which is a scientific question, but it's also an ethical question, and the ways in which it's one versus the other or both simultaneously is part of what I want to tease out today. Of course, the what are they question is a kind of experimental question, and the they is not singular. The they is quite heterogeneous if you look across the work in this field. And even in a given laboratory where the parameters of an experiment are moving from these to those in a given day or month or year and so forth. So I think the notion that there is a they to ask what are they about is belied by the extent to which it's a kind of a moving target. And yet, nevertheless, it has emerged as an apparently immensely important question, one that many have actually rushed to answer quite quickly. So this is just a handful of quotes pulled out of articles mostly from researchers in this area that have been produced over the last 3 or 4 months. And there is a kind of urgency to answering the what are they question. We must remember that these models are not actual human embryos. It would be dangerous to compare them directly to in vivo embryos, though of course in a certain sense that's the scientific project itself. These are neither embryos nor are we trying to make embryos actually, they are just models. Calling these synthetic embryos is rather misleading, even prejudicial. So the stakes seem to be quite high. In fact, it seems like the what are they question is really just code for, or slips very quickly into this question, what should they be called? What should they be named? And quite a lot of discussion has happened around this, even anticipating the scientific developments in this field. This discussion is at least 6 or 7 years old. Has been seen as an urgent discussion for at least that long. And here are just some of the names that have been offered, but also that have been controversial and contested. The one at the top of the list, which is also in the title of today's program, is perhaps the most controversial one that was boldly used in a paper last year by Jacob himself, I think, with purposes of a little bit of provocation. Thank you for that. And yet, I think that while we can have a debate about what these are and what these should be called, I think that it's important for us to, in fact, step back and ask, why is this the debate we're having? What's at stake in this debate? And one place to begin is asking the question, what power lies in bestowing a name? What's at stake in the act of naming? And let me suggest that there are many things that are at stake, and it's unequivocally an important question to understand these constructs that participate in the category of the human in some way, to understand what these constructs are and what their implications may be. But the focus on the problem of naming, it seems to me, is most fundamentally about the power to bestow the name and what power is being exercised. Again, the question is about the power or the exercise of authority, or what sort of flag is being put in the ground in the act of naming. And I think that the way to ask this question is to note the ways in which the idea is, the presumption is, that the terms of debate will be set in important respects by the name that gets used, right? And that the names that are being asserted in the name of expertise, in the name of scientific expertise. We might say in the name of scientific authority. There is a sense in which that act of naming is making a kind of an argument about how this debate should take place, about what terms are the important terms in this debate. What are the words that are going to be that are going to structure the kinds of questions we ask, the sorts of deliberations we engage in. And in this sense, I think it's important to appreciate that naming is a social act. It's a relational act, it's a social act, it's even a political act. If I'm not mistaken, we're in a country in which Elon Musk could not have named his kid R29647 or whatever the hell he named his kid. Because there is a sense in which the act of naming, which is the bringing of an individual into a community, naming them, you know, giving them a sort of singular identity, is recognized as an act which can be transgressive, which can violate the sort of norms of participation in a community. And the giving of a name, therefore, is an act of significance for a collective and not merely for the individual giving it. Indeed, the name creates the conditions for participation of the named individual, maybe, but also of the community in which that individual is named. And so, ditto for, you know, novel entities, the naming of that creates the conditions of possibility for collective discussion about it and its significance. And so, though one can think of this in a kind of trivial sense, that it is a political act, nevertheless, it's important to think about how that political act is understood and approached. And what I want to suggest is that the preoccupation with naming, the what is it and what should it be called questions, are a bad place to start this debate, that they are actually corrosive to the possibility for engaging in deliberation about what's at stake here. And I want to first try to illustrate this by pointing out the ways in which what's at stake here is not only the way the entities in the lab are talked about. Again, these are changing entities, these are not a fixed category, but also the ways in which the talking is talked about and thought about. So I want to take you back about 20 years to an actually quite similar debate that unfolded in the context of the emergence of stem cell research itself. Jacob was, at that phase, an early postdoc maybe in the Yanish lab, is that right? Maybe this is before that, okay. In particular, the sort of intersection of cloning, of the technique of somatic cell nuclear transfer, and the emerging domain of embryonic stem cell research. And so there was a kind of effort within elite members of the scientific community, particularly in the United States, to do away with a term that had come into being in the sort of late 90s, so-called therapeutic cloning, the production through somatic cell nuclear transfer of an embryo from which one would remove the inner cell mass and culture a stem cell line, to replace that term with this term, somatic cell nuclear transfer for the procurement of pluripotent stem cells. And the rationale for this was, this is more accurate. But the motivation for the concern was that cloning has a kind of negative connotation, that you imagine up the latest Arnold Schwarzenegger movie in which, you know, the subject of which is cloning or whatever. It's worth noting that that debate, which was ostensibly about the accuracy of terms, was really in effect about the sufficiency or insufficiency of public debate about this research area. So this is a comment piece from Science that declares that we must accept that democratic debate on cloning is bereft of any meaning. This is a kind of scientific evaluation of the legitimacy of democratic deliberation, which was significant internationally, and particularly in the United States at this moment, on the basis of a claim to know, to have the authority to name, to know what the correct name is and the incorrect name is, with the kind of thesis that if things are called by the wrong names, the meaning of those things, the conditions of possibility for talking about those things are perverted in some way, and democracy is thereby undermined. It becomes a kind of a fraud. And in fact, this campaign was taken to the halls of Congress. It played out in courtrooms across the United States around ballot measures in which there was an effort to prevent people from using the language of cloning in order to not, merely achieve accuracy, but to protect democracy from itself. That is a powerful intervention in a domain that is in the business of modulating power. It's a kind of intervention based on an authority that is itself not subject to democratic scrutiny or correction. That was the sort of basis for it. So the International Society for Stem Cell Research participated in this effort, and in 2004 sort of banned the term cloning in order to provide accurate, standardized terminology that will facilitate frank scientific, ethical, and public debate on stem cells and their potential for medicine. So I think what's worth seeing here is not just that a term was being changed, not just that there was some interest wrapped up with changing that term, fine. But what's important to see here is that an account of what good democracy and bad democracy is was getting advanced by a professional scientific society that positioned itself as playing custodian of public reason, in effect, of how people should talk about value-laden questions and issues in the domain of research, which this society participated in. So let me suggest that those problems, how a democracy should reason together, was of course a problem for democracy. And beginning the debate with the question, what is it and what should it be called, in effect subverts that process of thinking about what's at stake in asking those questions and whether indeed those are the right questions. And thereby undermines our capacity to contend with this important domain of scientific research and the ethical issues that it raises. And let me suggest something pretty simple-minded, and that is that a better question is this question: what limits do we need? And I think that this is an uncontroversial statement, that in this domain of research, where human biological material and the potencies associated with that biological stuff are being harnessed through techniques of bioengineering to channel it and guide it and nurture it and grow it in novel ways, that the human in some sense is at stake. That doesn't mean that the experiments being undertaken are in some sense a violation of the human. What it does mean is that the question of the human is present. And I would say that it's present in a material sense. How should we think about these entities and what is due to them, if anything, and under what circumstances? When is a kind of biological threshold passed such that we need to think about what's at stake here differently or approach it differently? But I would say it's also the human is at stake conceptually, in the sense that the circumstances require us to think and speak about the human in ways that are unfamiliar. And we can't simply straightforwardly reach into a sort of fixed definition and measure this domain of research against it. The ways of thinking about the human and the protection of the human and care for the human are unfolding in conjunction with the science in this domain, and indeed in other domains. And in a third sense, I would say the human is at stake. The ways in which we as human beings make moral sense together, the forms of political coexistence, the modes of reflection, the kind of exercise of moral responsibility that is incumbent upon us as community, as species, as polity, that the human is at stake in all of those dimensions and in ways that are interlinked. And so we have to think about whether these have the potential to be, these technologies of humanization, of rendering human formations, be they in synthetic human embryos or human embryo models or whatever you want to call them, or in domains like the production of human-animal chimera, the all sorts of arenas in which producing features of the human without producing humans is the scientific and technological project. That these have the potential to be dehumanizing, not merely in the sense that there could be a kind of transgression of the human, but that if we approach the questions associated with this in a way that seeks to avoid contending with the large questions that are back of them, that are somewhere on the horizon in the future, that we attenuate our capacity to ask questions about the ways in which human life is at stake. And that building that capacity in the face of this emerging domain of science and technology is absolutely crucial. So what kind of limits do we need? And there's a sense in which people who work in bioethics are often, accused of just sort of saying, no, you can't do this, it's setting stop signs or whatever. I think actually the most important role for bioethics is to think about how we think about this task of limits, because limits can open things up even as they can close them down. So the notion, and this is, I think, quite important, that knowing and naming the problem is a crucial act in the exercise of reflection and in the establishment of limits. That knowing and naming the problem is a precondition for asking what should be done about it. Well, if we think about this in terms of first knowing the scientific answers to questions like, what is it? And then and only then moving on to ethical deliberation about it, informed by, even constrained by those scientific answers, we put a lot of onus on science to put us in a place where we can have a discussion. In fact, if the answer to what is it is, well, it's really not known. I mean, that is a problem, is a scientific problem, it's an experimental problem. That should not stymie the ethical discussion, it should actually emancipate it from being preoccupied with the what is it question. I have to wrap up, but let me say just a quick word about a limit that is in a nearby domain of research that is a limit that's worth thinking about. So this is the so-called 14-day limit for human embryo research, which was sort of developed in the UK and has become a norm in many jurisdictions, not this one. It was only relatively recently that it became possible to experimentally transgress this limit. And at that moment, there was a kind of growing call for abandoning the limit, that sort of science had somehow rendered that limit obsolete. And indeed, that limit has been dropped by that same international society, the International Society for Stem Cell Research. But if we go back to the moment of genesis of that limit, the limit was less about whether there's a clear embryological rationale for drawing the line here versus there. The limit was about the ways in which the drawing of limits reflects a commitment of a society to restrain itself in the face of a broad recognition that there are dimensions, there are moral dimensions of this work that are important. There are worthy moral concerns. It was a witness to the existence of a moral idea of our society. This is from the Warnock Committee report, which became the basis for UK law around embryo research. That there must be some barriers that are not crossed, some limits fixed beyond which people must not be allowed to go. The very existence of morality depends upon it. So this is a very interesting comment from Thomas Aquinas. Human law has the nature of law insofar as it partakes of right reason, and it is clear that in this respect it is derived from the eternal law, but insofar as it deviates from reason, it is called an unjust law, and has the nature not of law, but of violence. That in effect, the natural law is the basis, needs to be the basis for human law. I took this picture during a coffee break in a discussion at Harvard. This is on the wall in the Harvard Law School. At a meeting at Harvard about the sort of problem of the 14-day rule and the ways in which organoid research, including embryoid research, was problematizing the kind of limit that rule established. I thought about, as I was taking this picture, the way in which the nature, which in Aquinas' imagination was the sort of source, the source of the eternal law, the creation, is a zone of ambiguity that we have to contend with in a new sort of way in a world where we no longer have recourse to that sort of imagination of the source and authority of law. And then, in fact, we have to look to ourselves. We have to recognize that the task of understanding, which is a political task, according to Hannah Arendt, is first and foremost, most fundamentally, a task of developing the capacity to think and speak about the things which we're able to do. And yet the ways in which the development of a kind of instrumental orientation to human life may be inimical to that capacity is something that warrants reflection in a profound sense. And so this is what I want to conclude with. The recognition that what's at stake in naming, what's at stake in the question, what is it, is not primarily the answer to that question, but the ways in which we make tacit commitments to making moral sense, to developing constitutive vocabularies that become the basis for undertaking the production of things in laboratories, among other domains, but also the sort of small c constitutional vocabularies, the ways in which we rearticulate what is the human that is in need of protection. And that these arenas of science and technology are not only arenas in which we have to think carefully about what should or shouldn't be done, but we have to think carefully about how we think and speak. Because what's at stake in them is not merely the experiments that are done, but the forms of moral imagination that we cultivate in ourselves or we fail to. Thank you.
Thank you very much for that point. What's in a name? We'll have a very brief panel discussion now and then open it up to all of us. What's in a name? Jacob, I'd like to ask you, you said it's a problem for yourself, but what are these entities for you? And maybe if you look at the discussions in your lab, what are they for the scientific community surrounding you?
You know, just one thought. I was listening to Ben at the moment. You know, what should we call them? And something that, you know, being living in Israel just made me think, like, I'm dealing enough with which political situation, one country, two countries, three countries, and then I made, oh, I didn't need this extra headache this month. In general, I want to say I'm personally agnostic to names. I think in the end, it's the most important is like if you use a term, really define what it is. However, I know that is not very practical often because, you know, you start a field and these people will understand what it is and they might think you are avoiding the certain answer. I really do think that in the end, it's a stem cell derived structure. I have no problem with calling synthetic. Some don't like the word synthetic, think it might suggest it's from plastic or it is sensitive. You know, people might think it's like sci-fi. You know, if that's what focus groups think, then I'm fine with it. But I don't see such a big deal about it. It in the end touches about, is it an embryo and is it not an embryo? And I think at the moment they are not, cannot be qualified an embryo because an embryo is about proving a potential, and that's in itself a problem. You know, they are embryo-like, they are embryo model. They may have the ability to become an embryo. And until that is not proven, then we have to acknowledge that it is unknown. As hard as it is, and as frustrating perhaps as it is, that's the way it is at the moment. In the end, I want to say I'm kind of fine with whatever expert societies people. I want to say, you know, that's not a big, the most important. I want to say fight for me to do on. I'm flexible about it, but most important in the end is just we have to really appear truthful to the public, not to have names that are very evasive, that are clearly not aware of the certain potentials we have.
I would have another question that is going more in maybe the sci-fi directions. It's very clear that naming it is tricky. Nevertheless, it has potential, and maybe the basic biology question cannot really be addressed if we can't compare the stem cell-derived embryo-like structure with the real embryo, so we can't validate the quality. Nevertheless, that material generates something that would be very valuable, let's say, in human therapies. So how do you see that? Where do you see the development of the thing, maybe even in the absence of a proper name, because I feel that this will require more development, and it will not only require more development. To see the potential, you will also need more time to look at what's happening. And well, these are too many questions. So question one, where do you see the potential and the development in more therapeutic use?
I think it's really actually in the case of human, when we say a model, the goal is not to necessarily have an exact replica of the human embryo, but of course it would make sense that we would want it as much, because you're trying to model processes, you want it. But it doesn't mean that it is useless if you don't reach those goals. So it's not about, in the end, the developmental potential, whether this entity can make an embryo. We need to have a system that has a reasonable efficiency, let's say at least 30, 40%. And we can perhaps reach later stages. And then we can really use this, I think, start thinking about using it for the applications I mentioned, really beyond whether the drug testing, whether genetic basis of malformations, and also, again, in the end, the most essential one is because we start with stem cells, we can knock out the stem cells and then observe which organs, what's going to happen developmentally. We can label them with fluorescence. Everything we've done with mouse genetics, my dream is the basic one, is trying to do this through this system. Of course, it's more complicated, but that is kind of the aspiration. Whether the other application that this system is not only teaching me which genes and which hormones are important to make a certain organ, but actually in itself, I'm basically, I can piggybacking on millions of years of evolution. Basically, I am unleashing the potential of naive pluripotent cells. My environment is very neutral, and they self-organize, basically, and based on that, you have a differentiation protocol. You can look at it as just a differentiation protocol that we are not the ones who are giving the cues, but actually use that this unfolds in itself. And I think the quality of the cells, we know it from mice, is much, much, much higher than you would get by 2D differentiation. Because I think it's almost, maybe it is impossible, differentiation in vivo, you cannot control when and which cytokine to give and the elasticity. And we know that you need the neighboring organs. We know that also the placenta influences differentiation. So this is really a complex differentiation protocol that might yield better cells. We don't know that, and that is what the science is going to explore, at least what I am going to explore.
One question connecting to that, you said the full potential or totipotent potential of human embryonic stem cells. Would you say that human embryonic stem cells are totipotent cells? If you say yes, it's the end of stem cell research in Germany, because then the Embryo Protection Act in Germany would protect stem cells as embryos.
I want to say, de facto, they are. So
hey, there's a little.
I mean, it's not in the sense that again, talking about terms and maybe needing to churn terms, not because of political reasons. I mean, I think they're not totipotent in the sense that they are a blastomere. Like blastomere is like the 8-cell, 16-cell stages. I guess there was a worry at the time being totipotent that if you from those you can make an entire embryo. I think that is the basis for this. They are, though, totipotent that you can, under cytokine or gene expression, they still have the potential, if you push them, to make these tissues. So it's a bit different. They're not totipotent in the sense of like a zygote or blastomere. They don't, but they have the potential to make the derivatives of it. And it may not be the same thing. And really, this is not a lawyer answer. It's just...
The lawyers will look at your answer, I'm sure. Just one final question before we then open up to the public. Ben, as a social scientist, would you say that this kind of research is able to change the imaginaries, the ideas, the concepts that we have of human reproduction?
Would I say?
Would you say?
Yeah, I mean, I think that the understanding of human reproduction has changed pretty profoundly over the course of the last, 45 years now, since the first child was born through IVF. And I think that the ways that if we look back over the history, the change has not merely been through the development of technologies, although that has been pretty profound, but also the ways we think about ourselves. We think about what the project of procreation is about and what norms we need to impose upon ourselves in order to ensure that it, coheres with ways of being human. And I think one of the things that's worth noting is the significant variation between, countries that are otherwise relatively similar in other regards, like say this one and mine, in how those norms have been articulated. Okay, maybe we're not similar countries. I could take that back. I saw some heads saw some people squint, around my university, there are advertisements for, women to make $5,000 for selling their eggs, whereas, it's prohibited in this country because there's a sense in which that's a transgression against, that violates some important boundaries that the human needs to be protected. Okay, there are things to learn from here. And the ways in which different societies have contended with these technological changes, I think, has also been a kind of arena in which societies, as they've been doing for centuries, have been giving articulation to, kind of what they care about vis-a-vis the human and how the human should be protected. I think that what's in the future is probably quite a lot more complex than what has come in the last 4 decades or so. So the challenges are significant. But that the best place to begin is not to begin with a technology or indeed with a biological entity, to ask in isolation, what is it? And does it have features that we need to worry about yet? Because if the answer is no, let's just not worry about them yet. Let's just keep going until we get somewhere that we're pretty sure the features exist such that we have to worry about them. I think a much better approach is to begin to look reflectively at the ways that we have contended with the questions that have emerged so far. And have we done well or not? I think there's a lot of room for improvement in a lot of societies. And one of the forms of improvement that I think is required is taking these questions out of the domain of a kind of esoteric technical discourse and making them human questions, making questions that are amenable to being addressed from the perspective of human experience, wisdom, and tradition. That's maybe an abstract statement, but I think there's a different kind of politics associated with that approach than one that focuses on a kind of technical questions first. And
point is clear. Thanks.
I think we wanted to involve the public, and we'll do that. It's hard for me to see who's raising his hand, but maybe you just start and you will be handed a microphone. 3rd row, no, 4th row, left.
Hello, good evening. Thanks a lot for the talks. They're very thought-opening. First, I have a relatively technical question to Jakub, which is, I was wondering if you try to take a fertilized egg from mouse and incubate it in your ex vivo or whatever you call it, incubators, and see how they would develop.
Yes.
So yeah, that's maybe I'll also ask the second question, which is to all, which is about whether the reason why we have so much discussion about the research on human embryos is related to the fact that many people think that we humans are special.
Oh. Okay.
That's all, thanks.
So the first question is indeed very relevant. As I mentioned, we go from day 5.5 until 11, and it's about 6 days. And indeed, we would like to cover a bit longer and a bit earlier. But actually, starting from a blastocyst is proven very, very difficult. We can grow them up to day 8.5 with less than 1% efficiency, and they're really abnormal. Versus what I showed you, we can go from 6.5 to 11, 60% really, really high quality embryos. And it seems for some reason that implantation, so what happens is like when you have a blastocyst, it implants. So that stage seems quite hard to recapitulate in vitro without delays, with very high quality. That is not to say that it's not possible, but at the moment, both in all species, that actually seems a problem. luckily, in a way, in the protocol that we do, I didn't get into the details, when we combine the different lineages, we actually bypass that stage. The first assemblies we see that are recognizable, they are post-blastocysts, they are post-implantation. So that is one thing we have now. But again, this could change. The second question about what the debate about human embryo research, and I mean obviously it is I can understand it's as Ben mentions about potential of life and what is life, and it is in the end it is justified. I mean if all these entities can become humans, then what do we do? But I don't think. On the other hand, it's not that this topic is like really new. I mean, we have been discussing what to do with excess of human embryos and with abortions and how many days can we grow them. I think we have like a new addition in a way now is that we have these entities. I would say simply the question is when do they become counted as embryo, if at all? Once they're counted as embryos, then there is regulation for that. But but then the complexity is how do you evaluate that? So I don't, and I want to say that really, the reason we want to do this, and we think that even though we don't know what we call it, we don't know what the potential, but I think it's very, very important that the public feels that nothing is being done in the dark, in the shadows, and actually start discussions very early. Even if actually maybe we'll not be able to progress, or they might not be relevant eventually. But it's very important to start early and hear the thoughts and define the questions and define the biological questions. And I think this was much, much sure. I don't think we're in a state like there's kind of a red flag, sound the alarm kind of discussion, but these things should be discussed, and it's also intellectually stimulating as well.
Does it work? Okay. Yeah, thanks a lot for your great lecture. So I was wondering, as a student of developmental biology at the University of Heidelberg, how much can we actually learn from your structures from the standpoint of developmental biology? Yes, of course, they look very similar to the natural embryo in many aspects. And yeah, the gene expression, the morphology closely recapitulates what we see in the natural case. But to confirm, we always have to go back, right? And if we really want to understand what is happening during development, we still have to rely on the natural embryo, I would presume. So yeah, what can we learn from your model? And even if it recapitulates development to 99%, we still would have to go back to the natural embryo for every discovery we make in your system. Or would that not apply after we have checked?
Yeah, no, it's a great question. I know it's a depressing answer that we've been working on this for 10 years and we've absolutely learned nothing mechanistically. But that doesn't mean it doesn't happen in the next 3 years. It's very. In the end, like now we're starting to see that produce, I want to say, of the systems. We do need to get the efficiencies higher. I think that is achievable. But I disagree with you eventually that we would always need to go back to human embryos. I mean, yes, there's going to be a level of characterization, and it's what's called benchmarking is for. And we will know how much similarities, and that's why it's very important. There are papers that there are very rare samples from abortions that can be used to make genomic data, expression data, and more of it of later stages. So we will know more what to control. But in the humans, they will never be an alternative. So this is as good as it gets, I would think. Is it good enough going to be? I don't know. And the other thing there is also by parallels, we can learn from non-human primates, which are also not very easy to study. But I want to say, because it's very, very early days, and maybe you're right, maybe it will not prove as a good. I think it will, but there is one way to find out, to do the research.
Thank you so much.
Yeah, okay. I would like to have a little bit more interaction between two of you, because you provoked this thought about how experts cannot just do the naming and then because that would be undemocratic. And then your response in the beginning of this debate was like, well, I think these are not embryos, but I leave it up for whatever people want to call it. Is this a kind of response that you think is favorable, and what is this the democratic response, and also what is a well-informed public debate in a democracy looks like?
Well, well, first of all, I'll point out that Jacob is here and having this discussion, which is not, you know, I mean, that doesn't go without saying. There are others who would not be so interested in participating in this discussion, I think. So I think that that's worth appreciating and applauding. Yeah, let's applaud. I think that let me kind of clarify something. And it's not that kind of giving the problem to the people makes it a democratic problem. It's a democratic problem from the start, right? Because it's how should we as, you know, how should political communities contend with these questions? What words should they use in that process, right? And why those words? So any decision-making, any kind of reliance on expertise is always a kind of political commitment. It's delegation of a question to those who are deemed by a society to be the most competent to contend with it, and deference to the answers that they give, right? But those are political acts and should be recognized as such. And if they are laid claim to, sort of in the cloning case that I was sketching for you, that's not delegated governance, that's appropriated governance. And we should see that, and we should appreciate it. And I think one of the risks in a domain like this is that matters of collective concern, I mean, these are matters of collective concern, right? There is the possibility of, in effect, appropriated governance, an inadvertent acquiescence to appropriated governance, because we think of the problem not as a problem of democratic judgment, but as a problem of, you know, expert assessment. And if we want to approach it that way, it should be done consciously. We should understand what it is that we're doing and why we're doing it. But it's not clear to me that when one steps back from problems like this, that beginning with the question, well, how much can you tell me, you know, to what degree can you answer these scientific questions that are really about experiments that haven't been done yet, you know, trajectories that the research may be on but hasn't been arrived at, the unknown in effect. Can you please give me answers to the unknown so that I can have a conversation about what's at stake in them, with the implication that I can't have that conversation until I have those answers, at which point the conversation is already, you know, way behind the 8 ball. That seems like a counterproductive approach. So what I mean by the sort of, I don't like to use the word democratization because it seems too simple-minded, but what I mean by seeking to engender the capacity to engage in robust deliberation about these things, it means thinking consciously about the ways in which responsibilities and burdens basically are placed on science to clarify things for us that are actually our responsibility to work out as a collective political community.
I just want to actually expand it, because it's like perhaps a lot about the naming itself, and I will talk more about the biology, which is going to dictate the ethics. I want to just kind of say, you know, it is generally assumed, I think, that, you know, as we scientists, we kind of like, already have an opinion, and I'm going out to the public to convince them of that opinion. But actually, many times it's not the case, that actually we are seeking support or guidance on certain things. I might have an initial opinion or dilemmas, but we are very open to get that feedback, because also perhaps the responsibility or some things I don't know. It's not about me, the scientist, and the ethicist. And it's very important for us in the end as well that the public, when it starts formulating its opinion, even in the end is very negative towards this kind of science, that it is done based on really being informed, fully informed, and not based on false assumptions and false propaganda. So that is, to me, is like very, very important.
Okay, thank you very much. I also don't know whom to ask. Going back to the naming, the point is in science communication, we tend always to think, okay, there's the public, the lay people, they won't understand, we should be careful, etc. But I would like to turn this a little bit around. Isn't it also, perhaps, do we also need some care how we name things? And so not in the sense of a professional split. A natural scientist is also someone who is part of our culture. And if one uses the term embryo, embryo is etymologically and in the common use related to the human body, to the human womb, with all the cultural connotations of motherhood, etc., etc. Is this a point, or is this just a kind of philosophical l'art pour l'art?
He gave it to me. Look, I mean, so first of all, good to see you, Christopher. I think that there is a risk of, this is about human development, which is to say it's about human reproduction, which, I mean, it's in the zone of reproduction. Not that that's its intent, but it's participating in a space of biology, which was never just a sort of pure space of biology divorced from the forms of relational and social and symbolic and legal and political and religious and economic meaning that are associated with them, right? And it seems odd that we should seek to ask questions about it in ways that ignore, occlude, or deny those dimensions, as though we can ask questions about it as though the lab is the only world and there is no world surrounding the lab. So I worry about that kind of orientation, a sort of seeking a kind of sterility in the discussion, which is a kind of funny word to use about a domain of the sort of reproductively related. But so I don't know if that sufficiently answers your question, but I think that we should not resist those things coming into the picture because they are in the picture. And we simply, it takes an act of force to deny their presence. They should be contended with because those things are in some sense at stake, but then it becomes a task of making sense of the ways they're at stake, whether they matter, how we make sense of them, so on and so forth. One of my concerns about what I've just called appropriated governance is not merely that there's a power grab and so on and so forth. I mean, that may be a concern, but it's also that, you know, that act of the International Society for Stem Cell Research in 2004 was an early step in a pattern that continues to today, which really is about trying to take steps in order to keep this conversation internal to the sciences. And actually, my sense, and Jacob can comment on this, is there's actually a lot more ambivalence and uncertainty when one is speaking to one's immediate community and colleagues, then one is speaking through the mouthpiece of the ISSCR, or when one is being disciplined by it when speaking to the media or whatever, right? And that should be, that uncertainty, that ambivalence, that should be an inclusive exercise. The doors should open, you know, let the rest of us into this conversation because there are things that are worth worrying about here, both that boundaries, that lines might be overstepped, but also that important opportunities to learn and to develop capacity that has the potential to benefit human beings might be foreclosed by not thinking carefully through these domains. But that requires a larger community than the small community that's hiding behind techniques to sort of shut down discussion.
There was another question, some rows behind there. Do you have a microphone?
Yep, I have a mic already. Yeah, thank you.
And there's another one here, please. But please start.
Yeah. Firstly, thank you for having this discussion. I think it's a rather magnificent hall to have this discussion in the first place. I can just stand up if you can't see me. So I think my first question sort of alludes to what my colleague already asked you previously, is that it's an exciting time to be doing research in biology, in developmental biology in particular. And in the past, let's say a month or two, there have been like 5, 6 models coming up in human development itself. And there have been a lot of scientific discussions, disagreements, and so on. But to play the devil's advocate again, at the end of the day, you still need human embryos to validate what exactly is a human embryo, what structure is actually referring to a human embryo. And in this end, do you think more countries should open their doors to human embryo research? And how do you think this is going to proceed further? with just models coming up, but with no validation whatsoever in vivo?
So it's a tough question, and I am, of course, a bit biased in my answer on this one. You're touching on two problems. One thing is, first of all, in general, I would say, although it's uncommon, but debates and discussions should be very, very welcome. I don't think we should be living in a world where Twitter is about, oh, how great your paper is, or just be silent about it. I think people should speak their minds, and I think scientists should be also receptive to critiques in general. There is, it's never a white or black thing. It's not, I think there's much confusion. I mean, if you make just a clump of ES cells and you have a primitive endoderm around them, and you just want to call them an embryo model, which clearly when you open an atlas, and even if you are very, very, let's say, drunk, there's absolutely no similarity, that doesn't make it an embryo model. It's like me saying, this is a table, because I want to call it a table, whereas it's a chair. So those easy situations are easy to weed out. We are doing all what we can. And you're right about maybe we are not able, never be 100% sure. But if you, maybe, if you have a model that gives you something that is very persistent, and maybe it will say, okay, I can then go back and go validate it in vivo in non-human primates. But you're never, and you shouldn't be never, be able to test this in human embryos in vivo. And it's just something we have to accept. And in the end, it is what do you learn from it eventually? Meaning, if I've learned gene X is really an inducer of liver differentiation, if I might never know whether this is real in utero embryo, but if I see in 2D differentiation that gene really gave me much more mature liver cells, which I could never get before, that is something. So we're never going to live in this world of absolute thing, but there are other benefits to look at. And honestly, also, we might not know. We don't know at the moment how this is going to shape. I also want to say still the knowledge about embryo at later stages. There are some labs really, for example, Dr. De Baker in the Netherlands. She's a radiologist. She's published a big paper in Science a couple years ago, and it's been doing really an atlas from abortions from week 4, 5, 6, whether it's through CT, whether now it's by spatial transcriptomics. So there is some knowledge, but we're never going to have this flexibility that you're talking about. This is absolute kind of, if I don't see it and do it in utero in humans, it's not worth it. We're somewhere in that, might be in that doubt, but I don't think that doubt is debilitating. But maybe in some cases, but this is the situation.
So we are committed to finish at 9, but we have 3 more questions. I like to collect them, and please make a very short point, and then you collect and include them into your brief final statements. Please, 1, 2, 3.
Thank you. So my question is, since there is a lot of diseases that are seen in the development of the embryo at later stages of the 14 days, and since this model is not labeled as an embryo, is there the possibility to push further the differentiation in order to continue this model to see other diseases at later stages?
Okay, please. It was your one. Okay, your question, please. Just speak. They will put it up. No. Speak into the microphone, please. Okay, then take mine.
Thank you so much. It's a question for Benjamin. You spoke several times about the difference in the legislatures of Germany and the US in regards to embryo research. As someone growing up in the US, studying in Germany now as a German, do you think, I'd like to know your opinion on the morals and the ethics of a German student going back to America to do that research, to do those experiments where it is allowed, and then returning back to a country where it is forbidden to publish that research and continue? What do you think about that?
Thank you very much. And there was another one here? Yes.
A technical one. So your lab produced these synthetic embryos. Are there other labs in the world who will now catch up, 10, 100, and A second one, they start from stem cells from some person. Is it only one person? So would this embryo then only have like one father? Could you make it 5 persons from which you mix it? Like how much flexibility you have here in the technicalities? Thanks.
Thank you very much. So please, Ben, if you start with a brief final remark, and then Jacob.
So I want to say 2 things. First of all, just because it can be done doesn't mean that it should be done. Not everything needs to be known. I mean, these things are things that are so banal that one feels like one shouldn't need to say them, and yet, you know, it's not absolutely essential that the embryo model be compared against the naturally produced human embryo if there is a sense that that would be a kind of ethical transgression, right? So many things that we could learn by transgressing fundamental ethical boundaries in all sorts of ways. And they would be really interesting, right? But we shouldn't do it. And then articulating those boundaries, of course, is the challenge and the task. The question about jurisdiction shopping in science is a really good question and a difficult question because science is an international enterprise. And in fact, that happens quite a bit, not maybe in the person moving from one place to the other, but in forming collaborations that move across jurisdictions where you do this piece there and that piece. I think that there is a risk to the ways in which that professional enterprise can with some ease slip through national boundaries, which tend to be the boundaries within which these kinds of questions get deliberated, just because they can get away with it. If science is to be in the service of society, it shouldn't seek the society where it can do what it wants the most easily. I think actually there needs to be a kind of commitment, I think as there is from not all, but some, to fostering deliberation around these difficult domains that unfolds at the level of the global. Because as I think, you know, as we've seen increasingly in the last decade, the CRISPR babies in China are a case in point. The problems that we're likely to contend with, particularly in domains where, you know, cell and developmental biology, meet bioengineering, meet reproductive technology, are going to be global developments that always play out in, you know, particular locales, particular communities, particular societies and countries. And we can't just say, well, you know, these nations do it this way and those nations do it that way, and we should forget about any kind of coherence. I mean, it's actually a challenge for humanity as such to foster the kind of reflection that is necessary to contend with these sorts of things. And that requires a genuinely cosmopolitan orientation to these sorts of problems. And science should lead in that way, not sort of subvert it.
Yeah, when the summary and the last 2 questions, we are in very, very early steps in the field, despite of the efforts and despite many, many great teams working on this. But then we have to be patient. We don't, we cannot model diseases yet in any of the models. But we have to understand where are these goals, because we're not just, you know, trying to make embryo models for the flashiness of it. That is the dream. We think it's not too far-fetched, but the road is very, very long until we reach that. We are trying to, of course, to reach the next step, and to reach day 21, which marks also the end of gastrulation in humans, and together with improving efficiency, and these things typically merge, because yes, we want to see the beginning of organ formation, and that is really, really becomes a black box. And in general, as I mentioned, because you could see that there are more, you know, we think we solve a question and then actually end up with 100 more questions, but that's actually good. That means that there's so much to ask, so much to explore. And that's why, you know, you asked about, we actually want to more labs to join our field. There are a lot of great labs already in it. I think there will be more labs. We are very open about, for example, you know, the device we make, we give it at no profit. It's the same cost. Everybody can get it for in academia for what I do, you know, whether it's training, whether it's sharing cell lines. We want this because I think this is a very, very big question. It's a very big challenge. It's bigger than than 50 labs, if you ask me. You need the engineering, you need the extracellular matrix, you need the biology, and I think that is what is exciting about the field. And I think we're going to see more of it in the next years, and we welcome that. And although I a little bit intentionally exaggerated when I answered to the question that we didn't learn nothing, because I wanted to kind of make the point that we need to be patient about it. But I think we have learned already a lot. The fact that we know the pluripotent cells that have that potential to make these tissues, that they have ability to make these structures, that you can get recapitulation of whatever, a lacuna or microvilli at these structures, it's already knowledge in itself for us, and it's just the beginning of it. And I don't kind of underestimate it just because I think as we're really in a good time in with genetics and easier genetics and models, and to really start dig deeper. But we're just really the tip of the iceberg. Thank you.
So when Torsten and I started the Marsilius project more than a year ago, the topic was lively participation, and it was bioethics. And I wouldn't have dreamt that we have this kind of interaction with these speakers at that very day. And I'm just super excited and pleased. And there is, of course, always a take-home message for you. You understood something, but the most important thing you should have understood is you are in charge of properly communicating your own science as well, to make that very clear and transparent. And we can't expect that society understands science if we are failing to communicate. And I think this was just a wonderful example, and I'm thrilled, and I think we should move over and get something to drink and eat and give our speakers a little rest before you come with your own questions. Thank you very much.